Understanding iron metabolism and its importance in relation to the brain is invaluable in monitoring the pathology of diseases like dementia. Although literature exists that link iron regulation to some mechanisms of neurodegeneration, more studies are needed to elucidate this relationship. Known as the most abundant transition metal within the brain, iron is responsible for a number of cellular operations, including neurotransmitter synthesis, oxygen transport, mitochondrial function, and myelination of neurons. As such, it is vital to cognitive function. Processes that involve iron use specialized mechanisms to manage iron in a tightly controlled manner. Otherwise, iron can be toxic and cause oxidative damage and induce diseases related to either its excess or deficiency. To achieve optimal levels of iron in the brain or brain-iron homeostasis, iron-modulating proteins control iron uptake, transport, and its storage. Understanding this phenomenon has been gleaned from similar metabolic mechanisms at the systemic level. Put simply, when the body ingests iron from food, an absorptive form of iron or heme and a non-absorptive form or non-heme results. Duodenal cells in the small intestine then absorb iron based on systemic iron needs. Its fate going forward can then be summarized as this: either stored in a non-toxic form, noting that Fe2 in the cell, if not managed properly, can produce free radicals, or iron is exported into the blood circulation. Once in the circulation and in its transportable form called Fe transferrin, different organs can uptake iron suited to their metabolic needs. The brain in particular acquires iron at the blood-brain barrier. This is mediated by transferrin receptors and divalent metal transporter pathways. Endothelial cells of this barrier then export iron into the brain interstitial fluid through a transmembrane protein called ferroportin, and iron is oxidized into Fe3. Iron is then either bound to transferrin or exists as non-transferrin bound iron. Those that are bound to transferrin are taken up by cells that provide support and insulation to axons in the central nervous system, and those that are not bound enter astrogelia, which are cells that provide metabolic support to neurons. Now, the nexus between iron metabolism and dementia remains controversial, in part due to dementia being complex. Dementia describes an array of disorders, as can be seen, affecting the brain that affects thinking, judgment, behavior, and the ability to perform daily tasks. Its symptoms range from memory loss, confusion, personality changes, and apathy and withdrawal. Now, recent understandings of iron metabolism in relation to dementia include these: iron overload, an idea continually expanded upon and known to cause damage to cells through free oxygen radical generation and in turn neuropathies characteristic of dementia. The level of neurotoxicity has been linked by Douglas to the dysfunction of ferritin, which is the protein responsible for iron storage. There has also been links to the absence of or mutations in ceruloplasmin, which is a protein involved in preventing oxygen radical formation. The resulting ceruloplasminemia has been studied in both mice and men to reveal the negative impact of mutant genotypes and genetic modifiers on iron balance. Roy and Andrew's study indeed confirms that as a result of this, patients often present with progressive dementia. They conclude, however, that more study is needed since multiple genes govern the iron distribution in the brain and nervous system, with which different neurological disorders may be associated. In 2014, Singh et al. Opined the transportable form of iron in the serum had a much higher buffering capacity under conditions of iron overload compared with cerebrospinal fluid. Accordingly, neuronal cells in the brain had an increased vulnerability to iron-induced toxicity. A newly defined iron-dependent form of cell death linked to dementia is ferroptosis. Here, neuronal death is driven by reactive forms of iron and associated lipid peroxides. An autopsy study of persons with dementia revealed neuronal plaques and tangles, particularly in the temporal lobe, responsible for memory, sensory input, and comprehension. This was consistent with Stockwell's study in 2017, which concluded that intracellular ions associated with these plaques, when it accumulates, induces the failure of an iron transporter protein, bringing the onset of Alzheimer's disease, the most common form of dementia. Now, while APP's role in other signaling pathways is not fully understood, this protein has been generally linked to ferroptosis insofar as cortical glutathione levels that affect its formation also coordinate ferroptosis. Yang and Zhang's study in 2020 confirmed that the hallmark of ferroptosis is the accumulation of these oxygen reactive species along with the reduction of glutathione levels, which causes redox dehomeostasis. Other recent studies also related the failures of the iron regulatory system to aging. Penke et al. In 2012 analysed MRI scans for iron deposits in subjects aged 11 to 72. They discussed how age-related iron retention serves as predictors of behavioural deficits, highlighting the decline in cognitive performance with older patients with dementia. Sheila Kumari in 2014 indeed correlated high iron levels to low behavioural and neurophysiological assessment scores. This was observed in another study as signposts particularly of Alzheimer's disease, but the point at which the iron rises in the pathogenesis remains unclear. Therefore, while these recent understandings have implicated iron as a biomarker for neuronal death and cognitive decline, there remain gaps in the evidence base. Future studies include elucidating the iron transport machinery at the blood-brain barrier, the interactions between various cell types and pathways regulating brain iron levels, and treatment options such as iron chelators that provide neuroprotection from brain iron dyshomeostasis associated with overload. In summary, what is known about iron metabolism in dementia needs to be refined. As suggested by Hosking's 2018 systematic review of literature relating to this very topic, more comprehensive and higher quality studies are needed to understand the role of iron in both cognitive decline and dementia.